In today's video, I'm going to be discussing the RTK-RAS pathway. RAS is a type of cellular non-viral oncogene whose common mutation is the source of many cancers due to a single amino acid from a change in a base pair of DNA. It is modified post-translationally where it is added to a lipid, more specifically the underside of the plasma membrane. RAS has a strong capability of binding GTP, hydrolyzing it to GDP. It can also serve as a molecular switch, flipping between an active and inactivated state. GTPase activating proteins, or GAPs, help activate RAS. GEFs, or guanine nucleotide exchange factors, can switch RAS from an inactivated state to an activated state by exchanging the guanine nucleotide that RAS is bound to. RAS is not normally oncogenic, but can become so through certain mutations. One example is changing the 12th amino acid valine to glycine. The RTK-RAS pathway is one example of how RAS can be activated. This pathway is activated by growth factors through steps: signal, reception, transduction, and response. In today's video, the specific response I'm going to be discussing is the upregulation of cyclin D. RTK, or receptor tyrosine kinase, is a type of enzyme-associated receptor that activates signaling pathways. In this pathway, receptor tyrosine kinases serve as transmembrane receptors. The portion outside of the cell binds to a ligand. The transmembrane keeps the receptor in the cell membrane, and the intracellular portion exhibits kinase activity. When RTKs do not have a specific ligand to bind, they are classified as monomers with an inactive kinase. A process called dimerization of RTKs allows for kinase activation. During this process, ligand binding brings two neighboring RTKs close together and allows them to dimerize. With the receptors interacting with one another, the kinase domains are brought in close proximity to each other. This close proximity allows them to cross and autophosphorylate. One kinase domain phosphorylates the activation loop of the other kinase and vice versa. Once this occurs, the kinase can now phosphorylate other proteins. The terminal phosphate of ATP is the phosphate that is used by the kinase, and when a kinase is inactive, the activation loop prevents ATP and substrate binding. The activation loop and ATP cannot be in the active site at the same time. This means that a kinase cannot phosphorylate its own activation loop, and if this activation comes into contact with an active site, ATP is not able to bind and phosphorylate to ADP. A type of protein called a scaffold can be used to bring together groups of signaling proteins. This proximity effect aids in both specificity and speed of signal propagation. The next steps of this signaling pathway are determined by the chemistry and shape of the intracellular portion. The next steps of this pathway involve domains, which are structures that exist on proteins. The two main domains that aid in the specificity of protein-protein interactions in this pathway are SH3 and SH2. SH3 bind to proline-rich regions in neighboring proteins, while SH2 bind to phosphorylated tyrosines due to domain recognition. Remember, phosphorylated tyrosines are found on newly dimerized RTKs. An example of a specific RTK is called EGFR. EGFRs dimerize auto and cross phosphorylating, which results in a new binding site for the adapter protein GRAB. GRAB contains an adapter with both SH2 and SH3 domains. SOS, a protein with proline regions, is able to bind to GRAB, and GRAB binds to RTKs. SOS, a type of GEF, is able to activate RAS to bring in a new GTP molecule. As previously stated in the video, GEFs activate GTPase proteins by switching inactive GDP to active GTP. Once RAS is bound to GDP, it is now active. GTP-bound RAS can now bind to other factors downstream. Now, the first thing this GT-bound RAS binds to is called RAF. RAF is a kind of mitogen-activated protein kinase, which are enzymes that phosphorylate proteins in response to mitogens. With RAF now activated, this can activate the next step in the pathway, MEK. With MEK activated, this in turn activates ERK. ERK can now be transported into the nucleus. Inside the nucleus, it will phosphorylate a multitude of transcriptional factors. One of the proteins that can be a transcriptional factor target is called MYC. MYC has numerous downstream effects, one of them being that it can lead to transcriptional upregulation of cyclin D. This is the G1 cyclin. This is significant because when upregulating this G1 cyclin, RAS and RTK signaling using ERK and MYC can lead to cyclin D and E2F proteins, allowing the cell to pass its proper restriction point. This is how mitogens enter the cell cycle. It is dangerous to have an overactive RTK-RAS signaling pathway because this is how overproliferation occurs. Looking at today's solutions, there has been more success with inhibitors of EGFR compared to RAS inhibition. EGFR inhibiting drugs contain tyrosine kinase inhibitors.